I would like to first give a brief introduction on the topic. My initial interest was to understand intracellular motility as indicated by this movie that shows endoplasmic reticulum ER streaming in an Arabidopsis or petiole cell. You can see the rapid movement of thick ER strands flowing just like a river. This streaming is driven by an actin-myosin cytoskeleton. A class of myosin found in plants is myosin 11, which is a plant-specific class of myosin motors. Among 13 members, we identified 11K as a major contributor to the ER streaming in vegetative organs. The process of generating higher order of myosin mutants, we found that plants defective of multiple myosin homologs showed aberrant bending in various organs. This phenotype is caused by a simultaneous knockout of myosins 11F and 11K. A sex-tuple mutant of myosins 11-1 to BFGK is the most severe mutant we have obtained. To investigate the defect of the double mutant, we tested phototropic responses by irradiation of unilateral blue light for 24 hours. The myosin 11FK mutant petioles showed enhanced bending compared to the wild type. A similar enhanced bending phenotype were also observed in response to gravity and in other elongating organs, including stems, hypocotyls, and roots. We suspect these mutants have lost its ability to straighten their organs. Hence, we explored the ability of these mutants to straighten their stem when subjected to unilateral gravity-stimulation. This is a two-dimensional clinostat that neutralizes gravity unilaterally. We place the plants horizontally for 45 minutes to initiate gravitropic bending, and then rotated the plants at 5 RPM in the dark condition. First, I will show you a behavior of wild-type stem. The stem bends upward in response to gravity, and then the bending stops and becomes straight. The straight posture is maintained for more than 10 hours on a clinostat. This result indicates that plants have a straightening system to restore their posture. In the case of myosin mutant, the stem continues to bend and coil during clino-rotation. This behavior indicates that myosin 11F11K mutant has a defect in the organ straightening to restore their posture. Based on this result, we would like to propose a scenario on how plants establish their posture. Environmental stimuli, such as gravity, as shown in this movie, induce organ bending. Plants restore their posture by organ straightening, probably after sensing curvature. We think that the organ straightening is a key step in establishing plant posture. It has been shown that actin and myosin 11 are required for the organ straightening. However, the substantial role and mechanism of organ strengthening remain largely unknown. In the next part, I'm going to talk about the role of the organ strengthening from a mechanical point of view. This part was carried out mainly by our collaborator, Dr. Satoru Tsugawa from Akita Prefecture University. Here is a strategy for investigating the mechanical role of organ strengthening. The movie shows gravitropic responses of the inflorescence stems of wild type and myosin 11 mutant. The mutant stem bent excessively compared to the wild type due to the defect in strengthening ability. The movement resembles that of someone who has difficulty with abdominal exercises. In the first step, we quantified the spatial-temporal bending behaviors of the stem by image analysis. To quantify when and where the stem bends and straightens, we defined a few characteristics. The time required to begin bending was shorter in the mutant. That is, the mutant stems started to bend before the wild type did. The initial condition of stem was approximately horizontal. We found that the position at which bending started, indicated by star, was closer to the tip in the mutant. This positional difference was cancelled off after 4 hours. This suggests that the elongating zone in the mutant stem was different from that in the wild type stem. To evaluate the degree of stem bending, we defined the inclination angle after 4 hours, which quantified the direction of stem tip. The angle for the wild type was nearly 0, while the angle for the mutant showed negative values. Based on these quantitative data, we hypothesized that force that prevents bending was compromised in the mutant. In the next step, we constructed a mechanical model using an elastic rod by axial elastic springs and angular elastic springs. The natural length of the axial elastic springs is assumed to be fixed based on data. The angular elastic springs are controlled by 2 parameters. First is gravitropic response, beta, and second is proprioceptive response, gamma, described in published article. Proprioception means the sense of self-movement, force, and body position. Here is the main results obtained our mathematical model. After making correspondence between model prediction and empirical data, we found that both parameters, beta and gamma, were lower in the mutant indicated by red. This means that not only prevention of bending by straightening, but also promotion of bending are weaker in the mutant. This was surprising for us because we did not expect the bending parameter beta to be lower in the mutant. We obtained data that could explain our modeling result. in the vertically grown stem, we can see more amyloplasts at the bottom of the endodermal cells in the 11FK mutant. In addition, average distance of amyloplast movement in 3 minutes was reduced in this mutant. These results suggest that the saltatory movements of the amyloplast are suppressed in the mutant. Since expression of 11K but not 11F was detected in the endodermal cells, this suggests that the 11K is mainly responsible for the amyloplast dynamics. We cannot distinguish between gravitropic and proprioceptive responses based on organ shape, but the modeling enabled us to evaluate the difference. Finally, mechanical tests of the stem were conducted using finite element method simulations. The results of the mechanical test are shown in red for higher stress and in blue in lower stress for lower stresses. I will show you stems after 4 hours of gravistimulation. In the wild type, the stress under gravitational force was higher at the supporting point. The principal directions were tensile stress at the upper flank, indicated by red, and compressive stress at the lower flank, indicated by blue, in the wild type stem. This is beneficial to the supporting point. On the other hand, in the mutant, highly stressed region was displaced from away from the supporting point. The middle region of the stem showed an inverted stress profile with tensile stress in the right flank, initially compressed region, and compressive stress in the left flank, initially tensioned region. The stress in the middle is much higher than that the supporting point. These results indicate that the stem shape of the mutant is mechanically unstable. Taken together, organ straightening adjusts the stress distribution to achieve a mechanically favorable shape. Having figured out the causative mechanical defects in the mouse mutant that explained the increased bending phenotype, we would like to know the underlying molecular mechanism of straightening. In the final part, I will talk about cells that require actin myosin function in organ straightening. Previously, we showed that the straightening defect in W mutant was significantly rescued by expressing myosin 11K under the control of myosin 11F promoter. This construct also rescued the kinked morphology of the mutant and implied that the 11F expressing cells have a key role in the straightening system. We therefore tried to identify the myosin 11F expressing cells. In the 11F promoter gas analysis, the inflorescence stems were stained from the tip to the base. In a cross-section of elongating stem, the gas signals were detected as circular distribution, including fiber cells and a subset of vascular cells. To determine which cell types are important for organ straightening, we used 2 promoters, 11F and IRX3, that have overlapping expression in fiber cells and vascular cells or xylem vessels respectively. We also employed WL promoter that expressed specifically in vascular cells. We produced transgenic lines that expressed myosin 11K GFP in the myosin mutant, or a dominant negative form of actin-8 protein fused to GFP in wild type, driven by each of the three promoters. Straightening ability of these transgenic plants were assessed by clinostat tests. First, we showed the results of a rescue experiment of myosin mutant. As you saw earlier, the 11FKW mutant stem continued to bend during clino rotation. The aberrant phenotype was rescued by expressing 11K under the control of WOL promoter, as well as 11F promoter. On the other hand, no rescue was seen in the IRX3 promoter plants as they continued to bend. In this slide, I will show you movement patterns of inflorescence stems on a clinostat. The patterns 1 and 2 indicate straightening behavior typically observed in wild-type stems, in which the bent shape gradually straightens during clino rotation, and in some cases becomes completely straight. Pattern 3 indicates a slight deficiency in the straightening ability of the stem, in which the stem maintains the bent shape at almost the same angle during clino rotation. And pattern 4 indicates a severe deficiency in the straightening ability of a stem, in which the stem continues to bend during clino rotation, and in some cases assumes a coiled shape. Of the 28 11F11KW mutant plants, 22 exhibited behavior classified as pattern 4. We analyzed 2 lines for each transgenic plant. Expression of 11K driven by promoters of 11F or WL reduced the frequency of plants exhibiting pattern 4 behavior. In contrast, expression of 11K by the IRX3 promoter did not significantly affect the behavior of the parental plants. Next, I would like to explain about the mutant actin-8 protein used in this study. Previously, Professor Morita's group has reported an actin-8 mutant named FRIZ1. Like myosin mutant, FRIZ1 has a defect in organ straightening. The mutant has a missense mutation from glutamic acid to lysine at position 272 of the actin-8 protein, resulting in a fragmentation of actin filaments. We therefore wondered whether this mutation could be used to inhibit the function of actin filaments in a site-specific manner. Hereafter, a mutant actin-8 protein will be referred to as Act8D. These are wild-type plants that expressed Act8D under the control of indicated promoters. When the 11F or WL promoter was used, aberrant coloring of leaves and aberrant bending of aberrant bending of stems and sleeks were observed. When IRX promoter was used, apical dominance was often inhibited, but the organs grew in a straight manner. Here is the results of clinostato tests of multiple plants. In wild type stem, only one of 34 exhibited behavior classified pattern 4. In contrast, most of the FRIZ1 mutant showed exhibited behavior classified as pattern 4. FRIZ1 mutant expresses ACT8D in most tissues of the inflorescence stems. Expression of ACT8D driven by 11F promoter or WL promoter increased the frequency of plants exhibiting pattern 4 behavior. On the other hand, expression of ACT8D driven by IRX3 promoter did not significantly affect the wild type behavior. Here, we summarize the results of cell type specific expression of myosin and ACT8D and their effect on straightening ability. These results suggest that cells expressing myosin 11F and WL play an important role in exerting straightening ability. To identify cells expressing 11F in the proximity of vascular cells, expression patterns were compared with various vascular and fiber cell markers, APL for phloem, SMXL5 and PXY for cambial cells, VND7 for xylem base cells, and NST3 for fiber cells. Plants expressing histone 4 GFP under the control of these promoters were crossed with plants expressing an RFP tagged nuclear marker under the control of the 11F promoter. We analyzed the expression by counting the number of fluorescently detected nuclei. Taking the image of this vertical section as an example, 28 nuclei were visualized by PXY promoter. On the other hand, 17 nuclei were detected by 11F promoter, all of which overlapped with the PXY promoter signals. The graph on the right shows the rate of signals that overlap each other. In the case of PXY promoter, about 60% nuclei overlapped with the 11F nuclei. And in the case of 11F promoter, 100% nuclei overlapped with the PXY signal. This slide summarizes the overlapping rate between 11F and each vascular marker. The results shows that the 11F signals partially overlapped with all of the markers. Among them, the 11F signals overlapped most frequently with the PXY signals and suggests that 11F is mainly expressed on the xylem side of the cambium. Finally, I would like to propose a model for establishing plant posture. Environmental stimuli that induce organ bending generates mechanical stress. The built-in straightening system then adjusts stress distribution, resulting in a mechanically favorable plant posture. Thus, plants may be constantly balancing their posture using straightening ability. In addition, this study shows that actin and myosin in 11F and WL expressing cells possibly in xylem side of cambial cells, is important for the straightening ability in plants. It is still an open question about the exact role of actin and myosin in the straightening system. I would like to thank my co-workers. Thank you for your attention.
Thank you for the presentation. Any other questions or comments?
Thank you very much for this very nice talk. I have a questions about the mechanical properties of the plant. Does it affect the mechanical properties, the mutants compared to the wild types?
We checked the mechanical strength of the viral type and the mutant stems, but until now, no difference. We checked by creep meter, push the stem by constant force to Newton, but deformation rate was not so different. And next, the composition of cell wall components, but not obvious difference.
Okay,
checked.
Thank you very much.
Thank you.
You had also these beautiful movements of the leaves when you turned your plants 90 degrees. The mutant kept bending the leaves towards the stems. Did you observe this phenomenon also then in these lines expressing dominant actin and
stem shape or leaf?
No, leaves. Because they were also like overbending. Qual type didn't do that.
Here?
If you can maybe play the not the movie where you follow the strengthening after 90-degree rotation.
Here?
And you had this movie, you know, while
movie. Sorry.
Yes.
Okay.
Yeah, when you had the slide, when there were these mechanical properties, the model, it was clear that it's not just the stem, but also the leaves kept moving towards the stem. So did you follow it then later on when you tried to figure out which tissues or which cell files are responsible?
I'm sorry.
Okay, we can discuss later.
Discuss later. I'm sorry. Thank you.
Any other questions? Okay, I have a question. In your model, the effect of gravity is incorporated to this. Is it right?
Yes.
So I wonder how the plants can know whether they are straight or not during the crinal rotation.
During crinal rotation.
For just the different growth of the cells at the bending zone is just stress, or I think your model is considering the effect of gravity, but the under crinal rotation condition, the gravity vector is nullified. So I wonder why.
At first, we placed the plants for 45 minutes to induce gravitropic bending, and after that, crinal rotation.
Yeah, but then the stems first bend towards the stimulated direction, but afterwards, the stem
maybe sensing curvature, they restore the posture, I think, but not
the effect of gravity.
Yeah.
So the time is over, and thanks for presentation.
Thank you very much.